So hypersensitivity reactions are basically exaggerated or inappropriate immune responses to otherwise harmless antigens, leading to tissue damage and various clinical manifestations. So the hypersensitivity reactions are basically divided into four types: type 1, type 2, type 3, and type 4. Now, this order from type 1 to type 4 reflects the relative speed at which these different types of hypersensitivity reactions occur, with type 1 being the fastest and type 4 being the slowest in onset and progression, while the type 2 and type 3 being moderately fast types. Type 1 hypersensitivity reaction is also called immediate hypersensitivity. Type 2 is also called cytotoxic hypersensitivity. Type 3, the immune complex mediated hypersensitivity, and type 4, the delayed form of hypersensitivity reactions. The term immediate highlights the fast onset of symptoms following exposure to the antigen or allergen in this case. Now, type 1 hypersensitivity reactions occur within minutes of exposure to the allergen. The term cytotoxic highlights the destructive nature of this immune response, where antibodies cause cell lysis, tissue damage, or loss of function by directly targeting and attacking the body's own cells. Type 2 hypersensitivity reactions usually occur within hours of exposure to the antigen. Type 3 hypersensitivity reactions are also known as immune complex mediated hypersensitivity reactions because they involve the formation of antigen-antibody complexes known as immune complexes. Type 3 hypersensitivity reactions also usually occur within hours of exposure to the antigen. Now, the fourth type, or the delayed hypersensitivity, is known delayed because this typically occurs 12 to 72 hours after exposure to the antigen. So let's take up the steps involved in the type 1 hypersensitivity reaction first. Now, the first step is activation of the immune system. So, for example, when your body encounters an allergen such as pollen or animal dander or certain food proteins, our system recognizes that antigen as foreign and potentially harmful. You can say that it is an exaggerated response by the body towards an antigen which is otherwise safe and quite normal in daily life. The next step is antigen presentation to T helper cells by antigen-presenting cells, or APCs. Now, this is an example of APC here, antigen-presenting cell. This could well be a dendritic cell or a macrophage, which come into play in this step. Now, these APCs, or dendritic cells in this case, are found in the skin, lymph nodes, spleen, tonsils. They're also found in the mucosal surfaces such as the lining of the respiratory tract, gastrointestinal tract, and genitourinary tract. Now, what is their role? They basically play a critical role in detecting and responding to inhaled or ingested allergens, pathogens, and other foreign substances. Now, these APCs grab hold of the antigen, or allergen in this case, and show it to other immune cells called T helper cells, also known as CD4 positive T cells. Since APCs present the allergen to the CD4 T cells or T helper cells, they are called antigen-presenting cells. So once the allergen is captured by the APC, it processes the antigen into smaller peptide fragments. This process involves the breaking down of the antigen into smaller pieces that can be recognized by the T cells in the next step. Now, what is this TCR? A TCR is a T cell receptor. It is a protein complex found on the surface of the T cell. The TCR is responsible for recognizing specific antigens presented by the antigen-presenting cells, or APCs, and initiates the immune response. Now, let me tell you why these T helper cells are also known as CD4 positive T cells. I assume you might have heard of CD4 positive cells before. Now, the question is why T helper cells are also known as CD4 positive T cells. The answer is that because T helper cells have a specific marker or protein on their surface called CD4, So basically, the positive symbol in CD4 indicates the presence of a surface protein called CD4 on the T cells. In this context, CD4 positive denotes that the T cells additionally express a CD4 protein on their surface. These CD4 act as co-receptors to the TCR in handling immune responses. Okay, now let's get back to the events in the type 1 hypersensitivity reaction. So what happens next? This response, or recognizing the antigen as potentially harmful, triggers the activation of certain immune cells, particularly B cells, which are type of white blood cells responsible for producing antibodies. Now, from our previous step, our T helper cell has been activated, and their activation means they will release certain cytokines or certain signaling molecules called interleukin 4, interleukin 5, and interleukin 13. These interleukins will then stimulate B cells to differentiate into plasma cells. Now, plasma cells are specialized cells that produce immunoglobulins or antibodies, and in this case they produce immunoglobulin E or the IgE antibodies. Now these IgE antibodies are uniquely designed to recognize and bind to the specific allergen that triggered their production. So in other words, I can say that your body can produce IgE antibodies specific to various allergens you have encountered. Now this takes us to the fourth step, which is recognition and binding to the allergen. So once produced, the IgE antibodies circulate in the bloodstream, ready to encounter their specific allergen again. So for example, when you are re-exposed to the same allergen, the IgE antibodies recognize it and bind to its surface. The binding of IgE antibodies to the allergen is a crucial step in the allergic response and sets the stage for subsequent reactions in the immune response. Let's see what is the role of mast cells and basophils in type 1 hypersensitivity reaction. For example, you have a re-exposure to the same allergen again. Now your body knows what is this allergen. Now these known allergen molecules will line up and bind to multiple IgE antibodies on the surface of mast cells or basophils. Now there is an IgE receptor, also known as Fc receptor, on the surface of the mast cells and basophils. Now the IgE antibodies form cross links with each other and bind to the Fc receptor on the surface of the mast cells or basophils. This cross-linking is a crucial step because it triggers a signaling cascade within these cells, leading to their activation and subsequent degranulation, which is associated with the release of certain inflammatory mediators such as histamine, leukotrienes, prostaglandins, and PAF or platelet activating factor. Now, what are the symptoms of type 1 hypersensitivity reaction? These inflammatory mediators cause characteristic symptoms of type 1 hypersensitivity reaction, or we may say symptoms of acute inflammation, including vasodilation, increased vascular permeability, smooth muscle contraction, mucus secretion, and recruitment of inflammatory cells. And the symptoms of this reaction may include itching, swelling, hives, bronchoconstriction, rhinorrhea, and in severe cases, anaphylaxis. So that's it for now. Thanks for watching.